Hi, my name is Sam Bakotich, and this is my final project for Introduction to Computational Chemistry and Biochemistry. For my project, instead of starting with the problem, I started with the protein. I explored what information was known about the protein and what information was missing. As someone with an interest in immunology, I focused my attention on the innate immune system, particularly on proteins that are involved with gut microbiome interactions. Eventually, I landed on NLRP6. NLRP6 is part of the NOD-like receptor, or NLR, family. It is a vital component of the innate immune system that is primarily expressed in the epithelial cells of the intestines, but there is evidence of NLRP6 activity in the lungs, kidneys, liver, and other parts of the human body. Importantly, NLRP6 is believed to assemble an inflammasome like the other members of the NLR family, but there is a lack of evidence to demonstrate the activation and mechanism for this inflammasome. NLRP6 is regulatory in nature, meaning that it helps to mitigate host pathogen interactions, and structurally, it's a leucine-rich protein made up almost entirely of alpha helices. As part of the innate immune system, NLRP6 is likely involved in a variety of human diseases. The NLRP6 inflammasome is important to regulating host defense during various types of infections. The inflammasome activates, causing a release of IL-18, which helps battle bacterial infections such as MRSA and Listeria, as well as parasitic infections such as those caused by schistosomiasis and Cryptosporidium. As early as 2012, researchers were investigating how to manipulate NLRP6 in order to combat some of these widespread bacterial and parasitic diseases. Although there is some debate surrounding its full impact, NLRP6 is likely involved in maintaining homeostasis in the human gut microbiome. The human gut microbiome encompasses the trillions of microbes that live inside human and gastrointestinal tract and are heavily involved in the functioning of the immune system. NLRP6 may play a central role in that relationship via its expression in epithelial cells, allowing it to be involved in the complex and poorly understood communication networks between the gut microbiome and the immune system. Poorly functioning microbiomes have been linked to diseases such as IBS and various autoimmune diseases, so it can be elucidated that NLRP6 might have a role in this as well. Another important regulatory function of NLRP6 in epithelial cells is the regulation of goblet cells. Goblet cells secrete layers of mucus throughout the intestines. Through this mucus, NLRP6 could help orchestrate large numbers of antimicrobial peptides, which help to limit the amount of gut bacteria and pathogens that can access the host cells. Although the mechanism is unknown, NLRP6 appears to be vital to the signaling and functioning of this pathway. There is a lot of current research happening around the NLR family. Most research cited in this project has come from the last 3 years, and the amount of research about this specific NLR is growing every year. Just to briefly summarize some of the research happening at a molecular level, there has been investigations into ATP binding and hydrolysis in inflammasome activation and how the domains in which this happens differs in different NLRs. Most of this research has been specific to NLRP3, and very little has been conducted towards NLRP6. There is also a debate on the structure of NLRP6, with 2 models appearing in the literature that are very different from each other. Cryo-EM and X-ray crystallography were used by Shen and colleagues to create a hollow cylindrical filament as seen in this project. However, Lang and colleagues have demonstrated a linear molecular platform when there is an LPS binding and an ATP stimulation. It should be noted that Shen and colleagues did not run their experiments with LPS binding and ATP stimulation. There are also pharmacology applications that are being investigated, as some small molecule inhibitors target the ATPase activity of various membranes of the NLR family. And finally, there's a lot of research going into the impact of NLRP6 on the gut microbiome composition. There are diverging paths here, with some studies saying there's just no difference in composition, and others stating that NLRP6 is vital. As I've said, the mechanism of NLRP6 isn't well understood. However, judging by the fact that it's been implicated in the involvement of human disease that impacts millions of people all over the world every year, I believe it's important to understand its function, structure, and mechanism better. In this project, I hope to use computational methodologies to provide more insight into how NLRP6 works inside the human body. One of the first choices that I had to make when deciding to focus on this protein was which model to use. The protein databank only had the Shen and colleagues model, so I didn't have access to the linear model proposed by Lang and colleagues. However, in the protein databank, there were two options: the full hollow cylindrical form or the PYD domain with an MBP protein fusion. I chose to use the filament model because it's a more stable version of the two, and because it seemed more interesting. I decided to use an approach that was inspired by thermal medicine when tackling this problem, meaning that I would manipulate the temperatures and compare the models from there. There is some current research involving manipulations of core temperatures as a mechanism for treating disease, and seeing as temperature is thought to be a modulator of the gut microbiome, I thought I might potentially get some interesting results. The first thing I did was download the filament file from the protein databank. Then I used the VMD plugin, AutoPSF, to create a PSF file to use in NAMD. 310 Kelvin is the standard for the human body, so I decided to minimize and equilibrate the protein at 300 Kelvin, 310 Kelvin, and 320 Kelvin for 10,000 steps, or 0.02 nanoseconds each. In order to do this, I had to modify a configuration file for each temperature. Instead of creating my own configuration file, I used one from the Ubiquitin water sphere tutorial, and just deleted the parameters related to the water sphere under the extra parameters section. I then found a charm parameter file to use for the simulation, and utilized an SBatch submission script to run it. The first time I ran it, I got a fatal error immediately, but I was able to fix this by turning MSM off in the configuration file. I then took the original PSF and the 3 DCD files from the various temperature runs and loaded them into VMD. At this point, I ran into issues with the filament being just too big to easily load, so I focused in on one chain and aligned them to the 310 Kelvin version by using the RMSD trajectory tool. During this, I analyzed the final frame from each temperature to visualize conformational changes between different temperatures. I also thought it would be interesting to run Gaussian for single-point energy on these files, so I used VMD to create a new PDB file for chain V, and then opened Avogadro to make the .com file for Gaussian. Unfortunately, chain V was too big and Avogadro crashed every time I opened it, so I focused in on residues 32 to 73, as this encompassed the second and third alpha helices. The second and third alpha helices are thought to be incredibly important for the formation of the filament, and the amount of residues here was the maximum amount I could get Avogadro to load properly. Unfortunately, what I didn't consider was that a molecule of this size is substantially more complex than what I had performed earlier in the semester, and Gaussian failed every time. One of the most interesting observations that came from the temperature manipulations was the conformational changes that happened in the 32 to 73 residue region. As you can see, at lower temperatures, there was an extra alpha helix formed in the loop region, where at higher temperatures, this alpha helix was extended another turn. I was curious about these conformational changes, so I decided to extend the computational modeling a bit more. I added 2 additional temperatures to run, 305 Kelvin and 315 Kelvin, and I ran all 5 runs for 1 nanosecond, or 500,000 steps. What's interesting about this photo, as messy as it is that the original PBD file is marked in orange, while the human body temperature, 310 Kelvin, is marked in teal, and there's huge outliers to both of those. So while I don't know the full implications of the modeling, I think it's fair to say that the protein's secondary structure is sensitive to temperature. To investigate the conformational changes more, I rotated the molecule so that the alpha helices were in front, but I could see the loop in the back. This was the easiest way to see the differences, what's happening in this region, without being blindsided. I should have been more careful when I took these rendered images, because the teal 310 Kelvin line is actually the same in both models. It just doesn't look that way. It's interesting because these pictures show that whenever the temperature is higher or lower than 310 Kelvin, there's a large variance in the loop region. I also went back to investigate the alpha helix formed at 300K with 10,000 steps, and found that when the simulation was run for a full nanosecond, the alpha helix was no longer there. I don't know exactly why this happened, but I imagine that it could have something to do with solvent effects, or any other limiting factor in my modeling experiment. I'm not familiar with the specifics of NAMD's implicit solvent functionality, but if this is simulating the effect of a protic solvent, that could explain why a longer equilibration eliminated the alpha helix. Finally, I made RMSD calculations at all 5 temperatures during the 1 nanosecond simulation that can be seen here. For some reason, 305 Kelvin is significantly lower than the other ones. Of note, the RMSD is very stable as the equilibration runs for longer timescales, which makes me think that this data is significant, even if I'm not positive of the implications. There are many steps that can be taken both computationally and experimentally in the future. To expand on what I did further, I could look at different solvents or different parameters to see how that impacts the protein at different temperatures as well. Additionally, when I first started this project, my grandiose idea was to find a way to make the protein function more efficiently by mutating it. I have a theory that I likely borrowed from someone else, that evolution doesn't always make the best version, but it makes the version that does what it needs to do at that time. I thought that if I could utilize a program such as PyRosetta to randomly assign mutations one by one and testing out many different combinations of mutations, I could be on the path towards finding a mutated version that functions better. And this led down a rabbit hole of trying to define what would make a protein function better, what would make the best protein for a human body. And apparently, it's a loaded question that I just don't have an answer to. I don't know if anybody has an answer to at this time. Unfortunately, 2 weeks was not long enough to learn Python well enough to get PyRosetta functioning, so I changed tracks. While I could have made mutations in VMD, I did not think that they would be random enough to simulate the fast-tracked evolution that I was trying to mimic. This approach was aimed at affecting the structure-function paradigm through mutation, but there are other ways to manipulate protein function besides altering structure, which led me to a thermomedicine-inspired approach. Another future step is creating better defined structure and functions for the NLRP6 protein. With the Shen versus Lang debate on cylindrical versus linear, that leaves a lot in the air as to how this protein would interact within a system. Investigating the binding sites and the behavior around those binding sites might lead to some more answers around structure as well. And finally, experimentally, almost all studies that have been conducted for this protein have been done in mice, with almost no human-based experiments and just very few mice experiments. Further experiments might give rise to more information about the structure and mechanism of the protein and its relationship with AMPs and other aspects of the gut microbiome. For the software I used in this program, or in this project, I used VMD, NAMD, Avogadro, Gaussian, PyRosetta, and agave. And finally, this is a complete list of the literature that I used to inform my research, including the original Shen and Lang papers, which can be found here. Thank you.